Science became important for me when I was in college. When I entered college, I really didn't have a focus in terms of what I was interested in professionally. But that is when I took my first course in biology, and then I was hooked. Nothing else mattered. I think I never realized how intricate physiology, or the biology of humans, was, and how elegant most of our body systems are, because that's what we were exposed to in the honors curriculum that I took in biochemistry and physiology when I was at McGill University. And then in addition, they gave us the chance to work in the laboratory. So you really had a chance to tackle a problem, come up with your ideas, discuss them with your mentors, and as I said, I was hooked. When I began my work, it was really at the end of medical school. It was then that I was exposed to the discipline of immunology, and I became aware of a real gap in what was known at that time. And the gap was, how do you initiate an immune response? So it was known that there were certain inducers of an immune response, particularly an infection. And it was known that the body could make a response to reject the infection. But what wasn't known was how the infection energized the immune system to make that response. The hypothesis was that the infection, or the antigen, the thing that was going to incite the immune system, had to be bound to the surface of a macrophage, a particular cell type in the immune system. And then that macrophage would display the intact infection to the immune system, and that somehow was necessary to get things going. So we tried to look for the capacity of macrophages to retain immunologically relevant information on its cell surface, and we couldn't succeed. So we then turned to another system. We turned to a system in which immunologists would take two sources of cells to generate an immune response. On the one hand, they took cells that are called lymphocytes that mediate the immune response, a fact that was discovered by Sir James Gowans in England about 10 years before I started my work. And then they would take another cell population that stuck to a glass or tissue culture surface. And by mixing the two of them together, they then could add an antigen or a stimulus and get an immune response. So I decided I would look at these adherent or sticky cells to see what their properties were, to try to understand how you initiate an immune response. And when I looked down the microscope, I saw something that was totally surprising. There were distinctive cells that were very tree-like or dendritic in their shape, from the Greek word dendron or dendrion for tree. And these cells had never been seen before. They didn't look like any other cell type. And then they were full of granules, which we characterized to be mitochondria, which are the energy supply machines for a cell. And in fact, these cells required a lot of energy because they were particularly motile. They would all the time be extending their processes and pulling them back in, much like a swimmer treading water, constantly extending and bringing back the extremities. And no other cell looked or behaved like this that had ever been seen. Most cells, when they move in a polarized way, in a very directional manner. But these cells were multi-directional, and we didn't understand what it meant, but in retrospect, it's a very appropriate type of movement, because what these cells do is they pick up infections, and they move in the body, and they embrace the immune cells that have to respond. So they really need their dendrites. A dendritic cell was a very appropriate term.